Okay. So for this lecture of MLT, QBI, we'll cover the shape analysis techniques. And so this feeds off quite well with what we've covered before with the segmentation lectures in how you actually take the things that you've segmented and start to do something useful with them. So again, there's quite a few literature and useful references. So we have sort of the morphometry with R book, and even though we don't really use R at all as a language, it's very similar to Python and MATLAB, and so it covers a lot of the ideas and how you do the measurement fairly well in R and what those measurements mean. The image processing handbook, as we've had before, and then a few smaller things on principal component analysis and shape tensors if you're interested in the details of that. So if we kind of look at sort of previously what we've covered, again, sort of the first lecture was all about image enhancement, or the third lecture. We covered data sets. We covered segmentation, how you can take images and divide them into sort of two or a finite number of classes. Some of the automatic methods that we have, so the hysteresis method, how you can use k-means on images, some sort of region of interest analysis, and a bit on sort of supervised methods and machine learning. And so when we look at kind of our learning objectives for today, it's sort of how do we quantify where and how big our objects are is kind of the first simplest point that we'll cover. So I think last week someone was asking about sort of measuring the radius of an object, and so this is the kind of the first thing we'll look into. Then the next part is how do we say something about the shape of the objects that we actually see? How do we compare objects of different sizes? And so if you have objects that are, some of them are large and some of them are small, but you want to compare the shape of them, how can you do that? How can you compare two images on the basis of shape sort of as calculated from the images? So how can you sort of say there's two different images, one's sort of before, one's after. How is this shape changed? Then how can we sort of take these objects and do some other sorts of analyses with them? So does anyone here do finite element modeling? 1, 2, 2.5? Okay. So that we'll cover a little bit about what steps need to take place in order to do that finite element simulation. Also, pretty renderings fall into the same area where you need to take your image, you've done a segmentation, and now you need to put it in this finite element or do something else with it. So in terms of kind of the outline, the motivation, we'll have some object characterization, volume, center, extents, anisotropy, and then we'll talk about sort of the shape tensor, which will be our primary tool for dealing with shape of sort of arbitrary objects, and sort of principal component analysis and how that fits together, ellipsoidal representation, some ideas about scale-free metrics, and anyways, that should get it all on the slide, yeah. Then anisotropy and oblateness, and so how that relates to this shape tensor, and then some of these ideas of meshing, which is quite important for the finite element simulations, and isosurfaces, and then something like surface area. And that will be the first part of the lecture. And so, we can look at what we've done, and we've sort of dramatically simplified our data, but we still sort of have way too much. And so when we take our original image, we have these sort of projections, reconstructions. We have this 56 gigabytes per sample that we started off with, for whatever reason. We then do this filter and enhancement step, and now it's 56 gigabytes of less noisy data. Then we do segmentation, and it's now 1.7 gigabytes. But it's still a huge amount of data, and it's more than we can really easily do something meaningful with. And if we go back to kind of what we wanted in the first place, we wanted sort of a single number, so sort of a volume of fraction, a cell count, average cell stretch, cell volume variability, and we don't have that. We still have billions of numbers we have to do something with. And so the first thing that's quite useful for us to do in order to kind of take this segmented image and start analyzing it is this idea of sort of component labeling. And so once we have sort of a segmented image, what we want to do is identify the subcomponents of the image. And so this is one of the most useful techniques that we actually have, where we take all of these segments. That if you look at, this is an example from a cityscape data set. So you can, I think, just go here. Yeah, so that link appears to be wrong. But you can go here, and this is a whole data set of cities and corresponding segmentations. And so pixel by pixel, what everything in the image is. And so you can go through these and kind of look at them. They're obviously very different than the images you're probably dealing with yourself, but it's very useful for kind of self-driving cars or similar sorts of applications. And so what we have here is an image, and we've done some sort of filtering and image enhancement, probably some sort of supervised approach to get this segmented image out. And so we now are able to identify cars inside the image. And the simplest kind of question we might have is how many cars are in the image? And how would you go about doing that? Any ideas? Yeah? Yeah, and how would you define simply connected? Yeah. Yeah. So you basically look at the images and you'd say if this pixel's touching other white pixels around it, then it's probably part of the same group. And so that's sort of what this component labeling idea is about, so that you sort of take the neighborhood of each object and you kind of grow it, and all the objects that are physically in touching each other when you look at these neighborhoods are in the same object. And so we have this function from a scikit image called label that does this for us. And basically the idea you have is you have sort of these 4 connected, as you mentioned, or 8 connected. And so that's basically if you consider just the points above, below, left, right to be part of a neighborhood, or if you consider sort of diagonal components as well. And, if you don't consider it, then anything that's touching just diagonally won't be considered connected. And if it is, you can consider them. And so basically, this does all of this for you. It has a number of examples of kind of if you have an image like this, and it's connected with 4 components, you get component 1, 2, 3. And if it's connected with 8, then you get all as part of the same component. So in a fairly simple image, you can see what it is. And so we can do that then quite easily on our image here, where we take our image and we just run label on it, and we now see that we've divided it up into each color representing a different car in our image. And now we can do something like a histogram, and we can see how many different objects we have. So that's sort of on this axis, the number of labels, and the y-axis is then how big each object is. So that we can see that some cars have thousands of pixels in them, and some of them just have a few pixels in them. And so when we look at the smaller ones, we might say that anything smaller than a certain size probably isn't a car anymore. So we could quite easily make a cutoff there and say the minimum size of a car is 100 pixels and remove some of these smaller objects from it. And so if we want to look at how this component labeling actually works, it's probably easier to start with sort of a simpler image. And so here we have a simple image where all pixels are either foreground 1 or background 0. And so that's what we spent all of the time with the segmentation lectures trying to do. is how do you take whatever sort of grayscale multivariate image you have and turn it into zeros and ones. And so here we sort of have a simple image where, you know, it's ones kind of on the diagonals and zeros everywhere else. And so the first thing we do in kind of a simple approach for this component labeling is to give each point in the image a unique label. And so with this, we just use a fairly simple rule where you say the color, if it's in the foreground, is equal to the x coordinate plus y times the width plus 1. So for those of you who've written lots of like C code or Java code, this is kind of like indexing through the array. And so basically, this guarantees that each index in your image, each pixel in your image has a unique index, and that no two pixels have the same index inside of them. And so what we get for this is now, you know, 1, 11, 21, 31, 33. And so it basically goes through the image this way. And once you get here, it now adds 8 or 9 to the offset. And that would be 10, 11, 12. And so you guarantee that each one has a unique ID. So there's many different ways you could do this. This is just sort of the simplest one you could follow, is by going through, and if it's in the image, give it a unique ID. And so what this allows us to do is set sort of the initial set of groups. And so all of these are now in their own groups, and now what we can do is go through it and try to combine those groups together. So in sort of a brush fire style algorithm, we kind of iteratively go through the image until it stops changing. And so it's called brush fire because you can sort of think of it like a fire propagation where, you know, if something's on fire, the things next to it will catch on fire, and then the things next to that will catch on fire, and then the things next to that, and then it sort of propagates out by neighbors. And so basically what we do is we go through each point in the image, and for each point, we go through each point in its neighborhood. And if the point is in the neighborhood, then the label of both points gets set to the smaller of the two numbers, and we repeat until no more labels have been changed. And so we can run through each iteration where it says, you know, for voxel 0, 8, it's 73, voxel 1, 7, or pixel 1, 7 is 65, and so they both become 65. Voxel 1, 1 is 11, voxel 0, 0 is 1, so they both become 1. And so that basically what we see is in the first iteration, these two now become the same, these two become the same, these two become the same, these two become the same. And then in the second iteration, these three are now the same, these three are now the same, these three are now the same. And so as we run this, you know, through iterations 3 and 4, it's really hard to see this purple against the background, but if you look at the slides yourself, you'll see it much clearer. But that you now have just 4 groups. And then if you continue, nothing changes. And so what this lets us know is that there's 4 unique groups in our image, and that those are sort of the IDs for them. So the fact that this is 1 and this is 49 are sort of irrelevant. This could also be 7. But the main point is that there are 4 groups when we're finished. And typically when you run this, after you finish, you clean up all the labels so that you have them sort of contiguously. So that you have 1, 2, 3, 4, or however you define it. And so we see that for 4 iterations, this task is complete. For obviously larger, much more complicated images with thousands of sort of crazy looking components, this can take much longer. And of course, there's much more efficient ways of doing this. So the way we had here was just a very simple approach, but this is the best way to probably understand what's going on. And so if we sort of plot this interactively, so we can sort of see as it goes through the iterations how the values in the image change. So that this 1 kind of propagates out from that upper corner, and this 49 propagates up with each iteration. And that in the simple image, after 4 iterations, we go from having 16 groups to having 4 groups at the end. So we can, of course, apply this to larger images. So we can take sort of our a small region of the car image that we had before and apply the same sort of thing. So where we give each index its own. So we take this small region where we had 2 cars, or a car divided in half, and we now have these 1s and these 0s. We give each point a unique ID, so 23, 24, 25, 38, 40, 41, and that we can run the same algorithm on it. And so we can now see that after 8 iterations, nothing changes anymore. This is the sort of really inefficient way of doing it, but that lets you kind of see what's changing and how many groups you have left. And then again, we can show that in sort of the same animated way we had before, where we see that these values on the top, because they're smaller, sort of propagate out with each iteration, and that we end up with only 2 groups at the end. And so that's how this algorithm, which is quite a useful tool to have, works for classifying these images for us. So now are able to take any image that we've segmented and apply this using the label command and get just the final outputs with the 2 labels that you have. And so we can also change the neighborhood. So we don't necessarily have to use the 4 or 8 connected that we mentioned before. You can use a 3x3 neighborhood or a 5x5 neighborhood. And if you use a 5x5 neighborhood, then it happens much faster. So you can see that it's finished in, 6 iterations, and that it goes much quicker. But we have the problem now that everything's in the same group because 5x5 was able to jump over this small gap. And so, if we go back to our original image, that might actually make sense because we do know that this is part of this, it's obvious for us that this is part of the same car. These aren't 2 different cars. And so maybe using 5x5 is a better way of defining groups because then it sort of jumps over this single line boundary. But if there's 2 cars that are very close together, then it would also jump over that boundary. And so deciding your neighborhood will affect how fast it runs, and it also affects what things end up getting grouped together. And so if we want these to be part of the same image, then 5x5 works great. If we don't want them to be, then we should use a smaller neighborhood. We can also use sort of a smaller kernel where we can take just the elements above, below, to the left, and to the right. And we can see that this goes much slower. So it takes many more iterations, and components like this don't ever get added. And so we end up having 3 groups in our final image rather than 2. And so depending on what you're trying to do, it would of course matter if this should be considered part of this image or not. And so if you're doing something with like poor flow rates in an image, and so a lot of, if you're looking at like geological images and you're looking how water would flow through, then you might say, 2 voxels or pixels that are only connected by a diagonal probably aren't actually connected, that water couldn't flow through that diagonal, that it needs to have a shared edge or a shared face in order for it to flow through. And so then you would want it to be counted as disconnected from this main pore. But for other applications, if you were looking at a cell and some of the things were very small anyways, then you wouldn't want to disconnect sort of one of the cilia just because it was offset by a slight amount. And so it really depends on what kind of problem you're looking at to if that makes sense as a criteria to separate things or not. Typically it doesn't, but there are certainly cases where it matters. And so, once we have all the connected components with the same label, we can start to kind of do more interesting analysis. And so with our original images, where we just had everything as black and white with 0 and 1s, we can't do very much with it because everything's grouped together. So we could do an analysis of all of the things. So something like volume fraction would be fairly easy. But if we wanted to know, how big are the objects, which ways are the objects pointing, whereas the center of the object, it's a lot more difficult if everything's grouped together. And so now we can look at each object individually and start to analyze it in more detail. And so if we go back to what we would like to do, so one of the first things was count the cells. So we've already gotten that taken care of because the number of components we have is the number of cells. And now we want to be able to sort of say something about them. And so we want to be able to compare the cells in this image to cells in another image. And, how do we start? And so the easiest thing we can do is sort of, do the center of volume. And so if we start off with, we keep just one of the labels in the image. And so we basically just say if it's in this label, it's 1, otherwise it's 0. We can now start to look at like where the center of mass or center of volume would be. And so here we can define the center by just sort of the x is the x coordinate, the mean x coordinate, y is the mean y coordinate, z is the mean z coordinate. And so we can take our image, put it in this sort of coordinate space where instead of having an image, we just have a list of x and y coordinates that are part of the object we're interested in. And then you just take the average of all the first components here and the average of all the second components here, and you now get sort of the center of volume being 5.16 and 7.77. 5.16 and 7.77 is here, right? Yeah, so we take this component being 1, and it's 5.6, so here, and 7.77, which is there. Which I guess, given it so much wider here, makes sense for the y component. We can then look at center of mass. So center of volume tells us where the center of the object is, and center of mass would tell us where the center of mass for that is. And so this is not something we can always do. It depends on what kind of images we've originally acquired. So with the color images we have before, so if we look at the original image for this, here, we can't really do center of mass because the pixel values in this image don't tell us anything about how heavy that pixel is likely to be. We can look at these and we can see that there's red lights and black paint and tires and things, but they don't really tell us much about the material composition of what's inside. Versus if we look at X-ray data, or X-ray tomography data, or we look at sort of electron microscopy data or some of these other forms, they give us a lot more information on the material properties, which means the pixel itself will tell us something about the density at that spot. And so when we look at images of cells, we can actually use the pixel intensity in order to calculate the center of mass, because for a lot of objects, the mass isn't distributed equally. If you have a cell, the nuclei is sort of much heavier, and then there's lots of empty space until the sort of cell membrane or cell wall if you're dealing with plants. But with an image like this, we have no indication of this. And also because it's sort of a 3D world projected on a 2D scene, even if we were able to say something about how this is concrete and it's probably, this heavy, we don't know how much concrete it is because up here it's a very small amount in 1 pixel, and down there it's a much bigger amount in 1 pixel. And so we'd have to somehow correct for that if we wanted to have a meaningful center of mass. But for cells, it makes sense. And so here we can sort of take a synthetic image where, we have this changing intensity. And so maybe this is the, electron density or, absorption amount at each point in the image. And we can calculate the center of volume and the center of mass. And so we can see the center of volume, so the kind of centroid of the object is right here. And the center of mass is then sort of shifted down and to the right because that's where sort of the heavier parts of the cell or object are. And so it's important to kind of keep these two terms separate. A lot of times they get mixed up and they have sort of very different meanings, and there's lots of images where looking at both of them is quite useful. And it also can be quite helpful for trying to figure out sort of the orientation of the object. And so if you had a square object like this, obviously there's not really an orientation to this square. But if you looked at the difference between the center of mass and the center of volume, you'd be able to say something about, like, the object's pointed this way. And so it can be helpful for that. Luckily for all of these things, you don't have to do kind of any of this tedious work yourself, where you have, taking all the coordinates in the image and then taking the average and then displaying them and everything else. That's already done for you in KNIME and Python. And so in Python, it's done with this tool called sort of region props. And so if you give it a labeled image and you give it an intensity image, it will go through, calculate all of the centers and calculate, the center of mass. I think it shows you down here everything it does. So it says the centroid. Euler number, extent, filled area, image, inertial time. So local centroid. Yeah, so it calculates all of these things for you. Yeah, and the weighted centroid. So the weighted centroid is the center of mass, and the centroid is the center of volume that we've talked about. Yeah.
In MATLAB, there is a function called the PW labeling,
Yeah.
which does basically the job of labeling the connected component.
Yeah.
Is that available as well, something like that?
Yeah, it's just called label in Python.
Right.
So we have that one up here. So the label, and this one's, you specify the neighborhood or connectivity, and then you get the results. And so this is just the scikit image.measure.label. So you don't, again, none of these things you have to implement yourself, but it's quite important to understand what they're doing and how their outputs are relevant for analyzing these aspects in images. And particularly understanding where they might make mistakes. That if you use sort of a for neighborhood, you'll have things like this disconnected. And so, your number of objects that you find could end up going way up or way down based on what neighborhood you use. And you should be aware that that matters. And particularly as you switch tools, that all of these parameters have different names in different tools. So I think, MATLAB or Python calls it connectivity. I think KNIME calls it something else. MATLAB probably calls it something slightly different. And so making sure you're getting consistent results when you use different tools is quite important. And that they're not just random parameters that you're setting, they actually affect your final results. But yeah, in general, you can sort of use these tools. You shouldn't rewrite any of this yourself. There's tools that do most of it for you. But kind of understanding the workflow and how everything fits together is quite important. And so if we look at what does region props do, we can give it our image and sort of have it print out everything that it finds. And so the first thing it tells us is the area. That's quite easy. It just counts the number of pixels. Then gives us a bounding box for the object, bounding box area, centroid, convex area. And so that's if you make sort of a convex contour around the object, how big is that? And so, because our objects are fairly square shaped, then the convex area is the same, but obviously there's plenty of objects where that would be very different. The actual coordinates in the object, eccentricity, the equivalent diameter. So this is then quite useful if you wanted to calculate the radius, because you could just take the equivalent diameter and divide by 2. The Euler number, extent, filled area, inertial tensor. So a lot of the things we won't get into immediately, but they can be quite helpful for doing kind of further analysis, that we get perimeter, orientation, all the weighted values. And so that then feeds in quite well to sort of the next thing that we're doing, where we look at sort of extents or caliper lengths. And so this is a bounding box, or there's a number of different terms for it, but basically what it tells you is sort of what is the extent of your object in X, Y, and Z if it's in 3D. And so we have kind of lots of information about each object, and so what this allows us to do is get some very simple measurements for what we're looking at. And so for some objects, it's quite useful. We can see that, taking an object like this, we can draw a bounding box around the limits and have sort of some idea of, the extents of this object in x and y. The problem for this is that it's not really the answer to any scientific question. So as we covered a little bit in the first lecture, the point of this course is to focus on how do we answer questions, not how do we develop tons of tools. And so no biologist ever asked, how long is the cell in the x direction? That's not a real question. Biologists might want to know how long the cell is, but they don't really care if it's in the x direction or the y direction. They just want to know how long it is. And so extents themselves aren't very helpful, but they give us sort of an easy representation for the image. And so here we can show, we have the simple bounding box, and we now can take our image, we can perform, so we have the segmentation, we can then perform the labeling, and then we can perform sort of a shape analysis on the image, where we show the centroid as sort of this dot, and we show the bounding box as sort of a box on top of it. And then we can do some kind of simple statistics on it, where we can take the area of each one, the bounding box, the centroid, the average color, and the label that we have for it. And then kind of show what each image looks like. And so we can see the first two are definitely cars, the third one as well. After that, it gets quite a bit harder to recognize. Looks like 5 and 6 are probably cars, but 7, 8, and 9, no idea if they're cars or not. We'd have to go back to the original image and look at it more carefully. And as we see, it's dividing things up into smaller pieces, and so of course that'll make it harder to recognize if there's actually a car in there or not. And so now what we can look at are sort of other techniques like anisotropy. And so when we have this box alone doesn't tell us very much. And what we're interested a lot of times is sort of what shape is this object. So that, for example, if we're looking at a car, we kind of know what a car's shape looks like. A car is kind of rectangular. It tends to be longer than it is tall. And so that would be a quite useful criteria for deciding if something's a car or if something's noise. That if it's a perfect square, it's probably not a car, and if it's elongated, it probably is. And so that's where we have tools like anisotropy, where we can look at how can we characterize what the shape is independent of how big the object is. Because we know, if the car is far away, it'll be very tiny. If it's close, it'll be very big. But the anisotropy should be about the same. And so what we can take here is we can sort of look, how do the dimensions compare to each other? And so there's, unfortunately, a huge number of different definitions of anisotropy. And so depending on what field you're in or which paper you happen to read, you can see, 4 or more definitions. But the basic idea is that we take sort of the longest side and compare it to the shortest side. And so if you're looking at a bounding box, this is quite easy. You just have both of your lengths. One of them's longer than the other. If they're the same length, then obviously it doesn't matter which one you pick. And that you now have some sort of ratio of these two values. And so here we have kind of these 4 simple definitions of anisotropy. And what we can show here is kind of how, as we make an object more and more elongated, so here we have an ellipse and we change how long the first axis is compared to the second axis, and we can show how the anisotropy changes. Shouldn't be that big of a graph. Well, so we can look at, did it stop playing? Oh, there it is. So that we can see the object get narrower and narrower and narrower, and we see in this first definition that the value kind of goes up and then it starts to explode. And so if we go to that first formula, we see, it's the longest side over the shortest side. And so as the shortest side gets close to 0, this value goes up to sort of infinite. And so this one could be useful, but it kind of explodes very quickly, so it's not necessarily a very stable result. if we look at sort of some of these, other ones, anisotropy number 2, starts off at 0 and then goes up to 1. And so kind of as you end up with an object here that's very, very elongated, you end up with a value that's fairly close to 1. And so that is a much nicer number to deal with because it doesn't explode, and you don't end up with a value of like 100 million for your anisotropy. That the highest your anisotropy can be is 1, and the lowest it can be is 0. And that, we have these other two metrics, which are also more well-behaved than the first one, but have sort of slightly different properties. And so the anisotropy number 3 also goes up to 1 and 4 as well. And so all of these, depending on what you're trying to do, might be more or better suited for your sort of problem. And the main thing is to kind of understand how the values change, because when you're averaging them together or looking at sort of statistics of a larger group, how the values change will affect it a lot. And so if you were to imagine a case where you had, 100 cells, and most of them were perfectly circular, but one of them was really, really elongated like this and had an anisotropy of, 1,000,000. If you were to look at the average anisotropy could be 1,000. When, of course, there was only one cell that was really, really anisotropic, and most of your cells were perfectly round. And so when you're picking these different metrics, you should be aware of how they allow the results to be skewed. And so maybe it's important that one of them was really, really anisotropic, and you want to have that in your result. But in many cases, you don't want one value that's really, really outlying to kind of skew the rest of your results. And so you'd probably prefer a method like 2 or 3, where the value is sort of more consistent, where it's either between 0 and 1, and if you have 1 that's really anisotropic, then it's just 1, and if you're averaging 1 with a bunch of zeros, then you still get a number fairly close to 0. So has everyone heard of principal component analysis before? Has anyone not heard of it? Just one? Or, well, just one brave enough, I guess, to say. Does anyone feel comfortable providing a definition or their understanding of principal component analysis? Yeah?
Basically, we're projecting your data set onto a basis that contains most of the variance in the data.
Yeah. And like a simple explanation for that?
If you think of like a 2D kind of scatter plot with random points, instead of having your x and y axis fixed, you'll align your axes to the direction in which the data is most varied.
Yeah.
And then we do that in n-dimensional space, and that's PCA.
Yep, exactly. So the problem I think with PCA is that your first definition was perfectly correct. That's what you learn in linear algebra class, but it's not very intuitive. Projecting a space, an n-dimensional space, onto something so that the most variance is along the first axis is exactly what PCA does, but it doesn't really help you know when PCA might be useful as a tool. And so basically, the second definition you provided was kind of much more useful because it allows you to actually think about it and visualize it a little bit better, because that's kind of what the tool is made to be used for, where you have sort of a very messy data set. And so example here would be sort of spectroscopy. And so for those of you familiar with Raman imaging or infrared imaging, you get a sort of spectra out. The units here are wave numbers. You don't really need to worry about that, but that you can kind of end up with, you have noise, fat, and protein. So you're looking at some kind of image of a cell, and you kind of know what your different spectra are. And now you have sort of a huge set of data, and you're trying to kind of do something with it. And so you could try just plotting, the first two components. And so here we have all of our test data, where we have these different curves, so protein, fat, and noise. And we could try plotting for all of the things that we're doing, the x and y coordinates, the first two values here for each one of the spectra. But that really wouldn't tell us very much. And so particularly when we're trying to visualize things in 2D, and that, this is not 4,000, I think it's 300 dimensional, because we have 300 different values in our spectra, it's quite difficult to visualize. And so what we can do here is we can perform Pca analysis, where we put the data in, and it finds of all these different components that we have, which ones are the most relevant for explaining the differences that we see in the groups. And so what we can do there is we can say, run the Pca analysis, and now we have a plot where the first component is here, and the second component is along there. And what's quite nice is we've now taken all of the very, very, very noisy data and sort of aligned it so that almost all of the variance is explained in sort of the first and second components, and that everything else is kind of noise. And so we've taken this very complex 300-dimensional data set and collapsed it down to just 2 dimensions, where we can now fairly easily see the differences between the groups that we were looking for. And so it allows us to kind of dramatically simplify what we're looking at, and it also gives us the ability to kind of look at the components themselves. And so we're able to see that the first component has some sort of spectra like this, and that the second component has a spectra like that. And so if we go back to our original thing, this was fat and this was protein. And so these two spectra, this allows us to kind of see how that breaks down over a very complicated problem. And that, of the data variance that we have in total, what is it? Over 50% is already explained by the first 2 components. And so we've dramatically reduced the amount of sort of variance that we have left. We can also do sort of another example with face analysis. And so we have this data set called the sort of Olivetti face collection, where you have all of these different faces. And this is what used to be done sort of before deep learning, where you can use Pca on all of the face components. And so you can basically take all the faces and turn them into kind of what are called eigenfaces, or the components for all the faces. And so you're able to explain which sort of components account for the biggest variance in your output image. And so, some people have sort of larger, darker eyes, larger noses, smaller noses, and so you can break down all the different faces, if they're centered and aligned, into this sort of eigenface representation, where you're able to represent all of the faces as sort of a combination of these different components. And so this is to kind of get back to real examples for us. What this is really useful for us for, or does anyone have an idea of why Pca might be useful for what we're looking at? In reference to sort of bounding boxes and looking at cells and orientation. So we can use it for that. So we can put in sort of the whole shape vector and do Pca to kind of like help us divide our 2 things into groups. So it's useful. That's something you can use it for. So whenever you have large sort of multivariate data sets, you can do that. What it's actually more useful for initially, or at least simpler, is for finding the orientation of an object. And so right now we were looking at bounding boxes along the sort of x-axis and the y-axis. And what we can use Pca for is to rotate the object we're looking at so that the longest axis is the first one. Because the axis with the most variance in the data is also the axis that is the longest in your image. And so if we take a simple image like this, we can calculate a Pca transform and have it rotated like that, and sort of show the first component is in this direction and the second component is in this direction. And so that if we apply this to, how do we not have it? I think it's this one. So if we take an image like this, we see that the bounding box is sort of that way. And if we put it into PCA, it will end up rotating the object so that this is flat along the axis and that we have the longest component in the first direction, which is a much better example than the one I have here, where it's a bit harder to see what exactly happens. But that you have this image and you're able to take this transformation and say, you know, these are the components and these are the orientations that you get out. So it basically gives us the longest dimension and then the next dimension, which allows us to measure things about our cell quite easily. And so again, this is not something we have to do ourselves. You know, the math for it you can look into if you're interested. But basically what we get is just these two components out of it that let us measure sort of how long is the cell and how wide is the cell, and give us a representation of the object in sort of these coordinates rather than x and y. And so what it lets us measure is something is sort of like what's the major axis length and what's the minor axis length for this object. And what we do this with is we sort of have this shape tensor where we take sort of all the voxel positions that we have. So we take all the positions in our image that are part of the group that we're looking at, and that we sort of decompose it into sort of eigenvectors and eigenvalues. And that is what makes up this sort of covariance. But don't really worry about what that means. That's just the kind of simple technique we use. That's what principal component looks like. So it's just taking this image that we have, or the shape tensor that we have, and breaking it down into eigenvectors and eigenvalues so that we can explain things like the shape, the major axes, and the dimensions. And so yeah, so you have access to all of these things. You don't have to ever really implement this yourself. You know, in MATLAB it's called printcomp. In Python it's called PCA. KNIME has it as PCA as well. And that you're able to put in your data set and it can kind of find correlations between the dimensions and give you the direction of largest variance, give you sort of an oriented bounding box for your image, and look at it quite usefully. And so I think we'll stop there. Are there any questions on the first part of this? Okay. So